Okay. Hello, everyone. Thank you for watching the review session. If you ask a question and you don't see it addressed in this review, please just check the description. I'm going to be putting more specific answers to questions there. So today I'm going to go over as much of the course as I can, starting with some necessary background and then getting into CRISPR-Cas and then the experiment itself and finishing off with an example. Because this is a lot of information, I'm going to be moving quite quickly. So to start off, the central dogma, as I'm sure you're all familiar, is that DNA encodes RNA, which encodes for protein. And the process by which DNA is converted into RNA is called transcription. And the process by which RNA is converted into protein is called translation. Now, it's really essential to know the different parts of a gene. Here I've put a basic gene diagram here where this blue region here is the promoter. And this contains sequences that transcription factors bind to and promote transcription of that specific gene. In red, I've put UTRs, or untranslated regions, this being 5' and this being 3'. So this would be here, this would be the 5' UTR, and this would be the 3' UTR. And then in purple, I'm putting exons. And then these black lines in between the exons represent introns. And then during transcription, when the mRNA is processed, the introns are removed, and transcription begins at the start of that UTR. So this would be the TSS, or transcriptional start site. And then what we end up with as mature RNA, this is 5', this is 3'. We still have the untranslated regions. And then this would be the coding sequence, which is just the combination of all these exons here with the removal of the introns. Then translation occurs, and the cell knows where to start and stop translation based off of the location of the start and stop codons. So all that purple region encodes for the amino acids within the protein, and the UTRs are not translated. Okay, so now I'm going to move on to just some worm biology. I'm not going to go over this. But worms are really useful, particularly because researchers are really interested in understanding the foundational mechanisms of biology, and it's really difficult to study foundational biology as it relates to human health and humans themselves. So we use model organisms so we can more easily study these foundational biology topics without the use of humans. So I'm going to list some of the pros of using worms as a model organism. First here, I'm showing a diagram of a hermaphrodite worm here on top, meaning it's able to produce its own sperm and eggs. And this is the vast majority of C. elegans. And then rarely males occur, and they're identifiable based off of the presence of that pointed tail here. Males are rare, and they arise through chromosomal non-disjunction in meiosis. So hermaphrodites have 2 copies of the X chromosome, which is the sex chromosome in worms, and males have 1 copy of the X chromosome. We write it as XO, meaning 1 copy and then nothing. So why use worms? Some of the key advantages of using worms as a model organism. The first is that their generation time is quite short. Meaning that if you put an egg on a plate, you can expect at 20 degrees Celsius for that egg to develop to the point where it's laying its own eggs in just 3 days. So we can measure the impact of genetic changes over many generations in a short amount of time. They're also transparent. And this means we can view all the tissues in their body without the use of fixation. So we can watch them grow and develop live. They also have a defined cell lineage. We know the cell lineage history of every cell in C. elegans, and it was that was determined simply just by watching the worm develop. And I think that's a really cool story. And particularly in developmental biology, that type of information is useful because we can see how um, conditions or different genetic changes can influence um, cell differentiation and tissue development. They also have quite easy maintenance. Um, Sorry, my handwriting sucks, and I also am not great at spelling, so bear with me. But the maintenance is pretty easy. We can maintain them in standard laboratory conditions on plates. They eat cultured E. coli, meaning they're easy to use. They're very powerful um, in developmental biology in particular. So that's something to keep in mind. Also, in this diagram here, you can see many different tissues within the worm that are similar to tissues within humans, including the muscle and the nervous system and the intestine and the germline. And I also want to just go a little bit further in detail about the germline itself. Um, I specifically study the germline, and a lot of people use C. elegans to study the germline because it's the majority of their body mass during the adult stage. And they have this really interesting germline morphology where the germline itself, there's two arms, and they kind of look like I have my arms right now, where these are the distal tips, and then each of them bends. And then I'll show you on the whole worm diagram where each germline is here. This is one. In this diagram, it's under some other tissues, so it might be difficult to see. And then this is the second one. And at the end of the germline, the proximal region, you have the development of fertilization and development of embryos within the uterus. Okay, so how this is relevant to our class is when we did the CRISPR injection, we injected the reagents directly into this region here. And we call this the syncytium. And that's because all of these germline nuclei are on the outside of the germline during this stage, and they share this cytoplasm. So if you put reagents within the cytoplasm of the syncytium, all of these germ nuclei on the edges are exposed to that material simultaneously, which is a huge advantage of working with the worm germline because we can do a single injection that exposes many germ nuclei to those reagents, and this is how we increase our odds of getting edited worms. Quickly, in this course, we talked about two different methods of worm maintenance. The first being picking or passaging, and the other being chunking. When you passage a worm, you use that metal pick, and this has advantages because it is very precise and you can pick exactly what stage in development and how many worms you would like to transfer. Chunking is easier, faster, and you can move a lot more worms at once. For genetics background, it'd be important to understand how to use Punnett squares and calculate the odds of getting what genetic makeup after a cross. I'm not going to go over that here, but I just wanted to say if that's something you have struggled with, there are many good tutorials online on how to use Punnett squares to solve the ratios of different genotypes in offspring. Worm nomenclature is very specific. Gene names tend to be the name of the mutant phenotype. So in our specific class, we're looking at a gene that, when mutated, leads the worms to be uncoordinated. So this is called UNC. And then it was the 32nd UNC to be discovered, so we call it UNC 32. Gene names are all lowercase italics, and how we denote different genotypes depends on where the different genotypes are in relation to each other. So I'm going to give some examples, calling this here. I'll describe what I've shown here. These are two homologous chromosomes, and this would be one locus, and this is a second locus, and they're on the same chromosome. This would be a different situation where we have two different loci here, and they're on separate chromosomes. So let's say this locus is gene 1, and this would be gene 2. And let's say this one's gene 3 and gene 4. So what if gene 1 and gene 2 are both homozygous mutants? Well, how would we write this? Well, because they're on the same chromosome, we could put a space between them. So we would write this genotype as gene 1 mutant, gene 2 mutant. And because it's homozygous, we don't need to write the second copy, because if you only write one copy, it's assumed the other homologous chromosome shares that same haplotype. Okay, so then in this 2 chromosome example, if these are both mutants, how would we write this? Well, different chromosomes are indicated by the use of a semicolon. So we would write this as gene 3 mutant, and then a semicolon indicating on a different chromosome, gene 4 mutant. Okay, so then what if gene 1 is heterozygous? How do we indicate that? Well, in this case, we would use, I need to write this a little smaller, so I'm going to erase it. In this case, because they're on the same chromosome, this would be written as gene 1 mutant, gene 2 mutant, and that is this chromosome's haplotype. And then I would use a slash to indicate on the homologous chromosome, and then I'd write gene 1 wild type and gene, I've run out of space, but it would be on the same line, gene 2 mutant. So I would use this slash to indicate this is what's on one chromosome and this is what's on the homologous chromosome. Now, in this scenario where they're on different chromosomes, what if gene 3 was heterozygous? How would we indicate that? So I would write this as gene 3 mutant and a slash to indicate on the homologous chromosome, gene 3 wild type. And then I would use a semicolon to show on a different chromosome. And then I'd write gene 4 mutant. Okay, does that make sense? Now, you may see in some of the lab materials the use of a double colon, and this means that 2 elements are fused together. So you can use this to say maybe you're using the promoter of one gene and the coding sequence of a different gene fused together in a transgene. You've seen that in this course when we write P-MEX5 driving the expression of Cas9. You've also seen that when we fuse 2 protein coding sequences together, in the case of when we say UNC 32 GFP. So double colon means a fusion, semicolon means different chromosome, and a slash means on a homologous chromosome. So now let's get into CRISPR-Cas. Um, Starting off, why do we want to edit genes? Well, we can induce many different changes if we can target a gene specifically at its endogenous locus. We can make mutations in that gene and see how they impact different biological processes. We can also add maybe a protein epitope tag, and this can be used to act for detection of the protein we're interested in. We can add something like a fluorescence protein, like GFP. And then by using imaging, we can see where our protein is expressed and maybe in what abundance. So for a long time, the field wanted to be able to specifically manipulate genes of interest, but it was very difficult to do in the past. Um, we have known for a while that a way to make targeted changes would be to exploit specific double-stranded break repair mechanisms in the cell. Here, this is just a schematic of two strands of DNA. And when that DNA is broken by a double-stranded break, there are two different types of repair that can occur. The first is non-homologous end joining, where basically the cell really doesn't like having double-stranded breaks. So if a accurate mechanism of fixing those double-stranded breaks is unavailable, the cell will basically just stick them back together. And this usually results in some form of misrepair, so short insertions or deletions, and we call these indels. So non-homologous end joining is much less accurate, but it's very quick, and it's better than having a double-stranded break. However, the one that scientists have been exploiting is this homologous recombination repair mechanism, by which the double-stranded break is stabilized by these single-stranded DNA binding proteins, and then based off of homology to the homologous chromosome, here this is a single-strand invasion. This is what's called a holiday junction, where this homology between this, in red, they're showing the homologous chromosome, and that single-stranded DNA that resulted from the double-stranded break leads to repair based off of the sequence of the homologous chromosome. So this is a way that the cell can make, that can fix a break based off of the sequence of the homologous chromosome, which is much more accurate and is less likely to cause short insertions or deletions. So I bring this up because imagine if instead of this homologous chromosome, we were to give the cell something that still had this homology to the single-stranded DNA that results from the double-stranded break. But what if we trick it into this region here, which it's copying from the homologous chromosome? What if we give it a fake homologous chromosome where we can add different DNA in this region between the double-stranded breaks? Then when it repairs based off of our fake homologous chromosome, which we call a repair template, it will induce our specific changes of interest, whether that be an insertion or some other desired alteration. So scientists have known for a while that if you induce a double-stranded break, it's quite straightforward to get the change you want by exploiting homology-dependent repair. So the issue was that how do we induce a double-stranded break? Well, there were mainly two different methods of inducing a specific double-stranded break. People had known how to induce double-stranded breaks in other regions of the genome, but to make one exactly where you want it to be, it required the development of these zinc finger proteins or talons, where you kind of like design a protein that is able to recognize your sequence of interest. And this took a lot of time and was very expensive. So for a long time, scientists were interested in finding a way that is cheap and quick to induce a specific double-stranded break. Now, simultaneously, other scientists are studying this weird sequence organization that's in bacteria. And they see that there's this really strange organization of DNA, where I have the laser pointer here, where there's these palindromic repeats, which generate these hairpin structures, and they're regularly distributed within this CRISPR array. And between them is these spacer regions, they call. So that's where CRISPR gets its name, clustered regularly interspersed short palindromic repeats, because that is what the CRISPR array in bacteria looks like. So it's this sequence oddity in bacteria, and scientists wanted to know what is its function. And they determined that once this CRISPR array is transcribed, it generates this pre-CRISPR RNA where you have these hairpin structures and the spacers here, which are the hairpin structures are able to be recognized by Cas9, and then generating these different complexes of Cas9 and these unique spacer sequences. And then they find that based off of these specific spacer sequences, a target DNA is cleaved. And one of the most interesting things about this CRISPR array in bacteria is that the spacer sequences, when they looked at them, they found the spacer sequences are very similar to that of the DNA sequences from viruses. Why does a bacteria have these regions of homology to viral DNA? And it turns out that the reason why they have it is as a defense mechanism. The Cas9 protein and this CRISPR RNA form a complex to target and disable invading viral DNA. So essentially, this weird sequence oddity in bacteria turned out to be this mechanism of bacterial immunity against invading viruses. And when this was discovered, what everyone was talking about is how this is a way that you can have a specific RNA sequence lead to a double-stranded break in a specific location of DNA. Maybe we could exploit this to induce a specific change in a DNA region of interest. And that's where the function of CRISPR-Cas in our experiment comes up. Now, I want to talk about the three main components of CRISPR-Cas itself that are required for the creation of a double-stranded break. First off being Cas9, and then the CRISPR RNA, and then the tracer RNA. The Cas9 is what induces the break. The CRISPR RNA is what provides the sequence specificity, and the tracer RNA is what links the CRISPR RNA to the Cas9. Cas9 recognizes the hairpin structure of the tracer RNA, and the tracer RNA has a region which is complementary to the CRISPR RNA here. So if I were to ask you to circle the CRISPR RNA and the tracer RNA in this diagram here, the CRISPR RNA is this whole region, the tracer RNA is this whole region. Okay. So if there's anything that's the most important for the exam, it would be to know and understand this diagram here, and how it applies to design of reagents for use in CRISPR-Cas9. The PAM sequence, which I haven't talked about yet, here has a sequence of NGG, N meaning any nucleotide. And the PAM sequence is what Cas9 recognizes and binds to. Cas9 can bind to any sequence containing a PAM, and this leads to unwinding of the about 20 nucleotides upstream of the PAM. And when I say upstream, I do mean on the 5' of the NGG PAMs. This would be 5', 3', 3', 5'. And then if the CRISPR RNA sequence here matches that region 20 nucleotides upstream of the PAM, then a double-stranded break is induced 3 nucleotides upstream of the PAM. Okay, so when designing an experiment to induce a specific change, you need to find a PAM that induces a cut closest to your desired insert site, and then design the CRISPR RNA 20 nucleotides upstream of the NGG PAM, and then finally design a repair template to induce your specific change. So the goal of our experiment this semester has been to add a GFP tag to the gene UNC32. Oops. And this generates a fusion protein where the GFP fluorescent protein is attached to the UNC32 protein, and we can use the GFP fluorescence to determine the expression pattern and localization of UNC32. So how do we determine where to tag the protein? So where do we insert that DNA sequence? This is just a copy of that same gene diagram that I showed before, where this would be the start and this would be the stop. Now, we want the GFP protein to be translated. We want it to be between the start and stop. Otherwise, it won't be translated. But if we put the GFP sequence in the middle of UNC32, we might disrupt the UNC32 protein folding. So the safest place to start when first designing a tagged version of a protein is to either put your sequence directly after the start, so directly 3' of the start codon, or directly before the stop, so immediately 5' of the stop codon. And then you want to pick a PAM that induces a break as close as possible to the insert site. So in this sequence here, I'm just showing a start codon and some generic surrounding sequence. This is similar to UNC32. We would want the insert site to be directly 3' of the start codon, so in between these two Gs. Now, if this is all you were given, you didn't know any of the surrounding sequences, you could see this and go, well, that's a PAM. PAM sequences are NGG. And where would this PAM lead to a cut being induced? Well, it would be 3 nucleotides upstream of the PAM. Here I've just put N's to mean generic, could be anything, nucleotides. And then when you're designing the CRISPR RNA sequence, it would be the 20 nucleotides upstream of the PAM. So everything encompassed here, and then additional bases, which are not shown. So for an NGG PAM, the CRISPR RNA sequence is the 20 nucleotides upstream. I'm just going to show the CRISPR diagram again, because it can be confusing why the CRISPR RNA sequence is the exact same as the 20 nucleotides upstream of the PAM. So if this is your PAM here, the 20 nucleotides upstream of the PAM, this would be the sequence. Okay, Now we know the CRISPR RNA is complementary to the opposite strand of the PAM. So the strand opposite, the 20 nucleotides on the other strand of the PAM. So if the CRISPR RNA is complementary to the reverse complement of the 20 nucleotides upstream of the PAM, that's the same as being the same sequence. So this here will be the same sequence as these 20 nucleotides here. So if we're looking at the strand that has the NGG PAM, then the CRISPR RNA sequence will just be the same as the 20 nucleotides upstream. So this can get a bit more complicated when we're looking at the other strand, so a CCN PAM. I'm going to move this down so I can. So I'm going to change this to what if this was the sequence? So now, still we want the insert, let me just read. Still we want the insert site to be directly 3' of the start. But in this case, our PAM is the CCN PAM. See, these are all arbitrary nucleotides. Then where does this PAM induce a cut? I'm going to say this first and then I'll explain it. 3 nucleotides downstream of the CCN PAM. So this would be where the cut site would be, which is still pretty close to the insert site. And then the sequence of the CRISPR RNA would be the 20 nucleotides downstream of the CCN PAM, and it would be the reverse complement. So reverse complement of 20 nucleotides downstream. Okay, I'm going to write that as a rule here. For the CCN PAM, the CRISPR RNA is the 20 nucleotides downstream. That this would be the reverse complement. Okay, to explain this, I'm going to draw out the other strand of DNA. This is 5', this is 3', and then the other strand is in the opposite direction. So this is the complement of that strand. And if remember, when we write DNA sequences, we always write it from 5' to 3'. So if I wanted to write out this strand of DNA, I'm just gonna rewrite it. It's exactly the same. Just I'm gonna write it 5' to 3'. How many ends? 1, 2, 3, 4, 5, 6. Okay, 1, 2, 3, 4, 5, 6. G, C, G, G, C, A, T, and then 3 ends. Okay, so this is the same thing as this strand here, but just written again the way we're used to reading DNA. And looking at this strand, so the strand opposite of this starting DNA sequence that has the CCN PAM, we can pretty easily see this would be the PAM here, NGG, and the cut would be into 3 nucleotides upstream here. And then the CRISPR RNA sequence would be the same as these ends here, so 20 nucleotides upstream. Okay, now I'm going to remind you here on this black starting sequence here, how we determined the CRISPR RNA using the CCN PAM. we said this would be the CCN PAM right here, CCN. The cut would be induced 3 nucleotides upstream, which is the same as what we see when we rewrite that strand, that opposite strand, to look at the NGG PAM. And then we said the CRISPR RNA would be the reverse complement of this region, which would be the same as this region here. Okay, I hope that makes sense. So now that we know where we want our insert to be, which PAM we're going to use, and we've designed the CRISPR RNA, we need to design the repair template, which tells the cell how we want it to repair that double-stranded break. The repair template is pretty much 60 nucleotides on each side of that proposed insert site. But also, one thing that we do is we mutate the PAM. And we try to do this in a way that doesn't disrupt the gene. So if our PAM is in the coding sequence, then we make sure it doesn't make a mutation in the protein sequence itself. And if our PAM is in a non-coding region, we might make sure that the mutation that we're inducing doesn't change well-conserved regions within the UTRs. I want to highlight that it doesn't matter if your PAM or CRISPR RNA are in coding regions, non-coding regions. The only thing that matters is that it puts a cut as close as possible to the insert site. So when we design the repair template, we try to mutate the PAM if this is not possible, meaning any change that we would make to the PAM would disrupt the amino acid sequence of the protein. Then we may choose to mutate the sequence of the CRISPR RNA to prevent binding. So we do this because if the PAM or CRISPR RNA are changed, then Cas9 cannot continue to recognize and cut that same region of DNA, which might result in unwanted mutations. So we design those changes. We also design where we want the insert, and then we do 60 nucleotides of homology from the most recent change. 60 nucleotides homology from most 5' and 3' edit. Okay, so that serves to tell the cell, hey, this sequence we've given you is the homologous chromosome. And then it'll trust that when it repairs the double-stranded break with our inserted DNA sequence, that it's using an accurate homologous chromosome, even though we've essentially tricked the cell. Okay. So now I'm going to describe the injection and the experiment that we did within this course. The first thing we did was we injected, we used this protocol from the paper that you guys have been doing journal clubs on, where the starting strain expresses Cas9. And this is really useful because when we want the Cas9 to be present in the germline, and one method of getting Cas9 into the germline involves purifying out Cas9 protein and then mixing it with the RNA in a tube, and that can be kind of finicky and is also kind of expensive. So in the paper that you guys read, they have the worms express Cas9 in the germline. So instead of injecting Cas9, the Cas9's already there. And also, they have the worms mutated for the UNC119 gene. So they don't have functioning UNC119, meaning that they show the uncoordinated phenotype. This can be confusing in this course because UNC119, the phenotype goes back and forth throughout this experiment. Our worms are UNC, then they're non-UNC, then they're UNC again, and then they're non-UNC again. So why are we doing this? We use UNC119 as a tool to more easily see the changes in genotype without looking at the DNA itself. So what I mean by that is that the worm that we inject, the genotype of that worm, it has the UNC119 mutant. It has PMEX5 driving expression of Cas9. It also has PHSP, I don't remember which HSP it is, driving expression of CRE, recombinase, and then an orange fluorescent protein. So these worms are uncoordinated. When we inject them, they're UNC. So the phenotype is UNC. Now, usually when we're trying to induce, Usually when we're trying to induce a large insertion, the odds of getting that insertion are quite small. So being able to see which worms are most likely to have the insertion is extremely useful. So this paper suggests putting within the GFP a sequence of a wild type UNC119. And therefore, if you get the GFP insert into the genome, then you will also have expression of UNC119. Now, this expression is from a different locus than the wild type UNC119 locus, which is still mutated. But because we've rescued it alongside our GFP, now the worms have expression of wild type UNC119 and are no longer uncoordinated. This ties the phenotype of non-UNC worms to the presence of our GFP insert. And this allows us to see which worms are most likely to have that insert without having to license genotype hundreds and hundreds of worms, which is usually what people do when they're trying to insert a large region of DNA. So after the injection, if we have a successful insertion of the sequence, the genotype would be UNC32 GFP with that UNC119 rescue gene. And on a different chromosome, actually, I think in this one it's on the same chromosome, so I'll do just a space. We have PMEX5, Cas9, so now if the insertion is integrated, then we go from the parent being UNC to now having progeny, which are non-UNC. So now we confirm the presence of this insert by genotyping. And I want to describe genotyping because it will be important to understand how you would design primers for a genotyping experiment. So let's say this is my wild type. To be wild type. This is your favorite gene. Where I put lines, I'm putting where we would want to put the insert. Then if we have a successful insert here, we'll just call this UNC32 GFP. If this is a GFP tag at both chromosomes, would have its insert. I'm making the insert in purple. So how can you differentiate between these two possibilities? Wild type UNC32 and UNC32 with the GFP insertion. Well, we design our primers to be 5' of the insert site and 3' of the insert site. So if you were to do a PCR amplifying with these primers, well, actually in this case it would be primers on both of the chromosomes, but let's say that this amplified region between these two primers is like 200 base pairs. What would that look like on a gel? Let's say that's what that looks like. Now, if we have 2 copies of that GFP insert, now you still have primer binding. It would be 5' and 3' of the insert. And then when the DNA between them is amplified, it would be a much longer region. If our insert's like 500 base pairs, then it would be 700 base pairs in the genotyping PCRs. This would be and that would look like one larger band. What would you expect if you were looking at a heterozygous worm? So one copy of wild type and one copy with the insert. So you would have the primers annealing on both chromosomes, and the amplification between them would be both the 200 base pair product and the 700 base pair product. And when you were in the gel, you would see the presence of the heavy band and the light band. Okay, so when you're designing a genotyping primer set, you want to use a primer that's 5' of the insert site, and then you want to use a primer that's 3' of the insert site. One of them in the forward direction, and then the reverse primer would be the reverse complement. So now, after verifying the genotype by doing the lysis and genotyping, then the next thing we want to do is, remember, our final goal is just to have UNC32 GFP. So we want to remove everything else that's in this genotype. And the first thing we do to try and get this as close as we can to UNC32 GFP is to remove this UNC119 rescue. So remember, its presence is only to help us identify the worms which are most likely to have the GFP insert. Now that we know we have the insert, we want to get rid of it. The UNC119 rescue is between 2 LOX-P sites, and those can be, the region between 2 LOX-P sites can be excised by the action of Cre recombinase. Now, we have this transgene, which has the promoter for a heat shock protein. And heat shock proteins are expressed in response to high heat, driving expression of Cre recombinase. So when these worms are exposed to high heat, they'll express that Cre recombinase because it's under control of the HSP promoter. And this recombinase will act on the LOX-P sites, which are around the UNC119 rescue gene. So if the heat shock works, our next genotype would be UNC32 GFP, now no longer with the rescue of UNC119 because it's been excised by the Cre recombinase. And then UNC119, which is still mutant at its endogenous locus, and then PMX5, Cas9, Okay, so now the phenotype, because it's lost the rescue of UNC19, is now back to being uncoordinated. So we know if the heat shock works based off of if the progeny of the heat shock worm are uncoordinated. And finally, how do we remove all of this stuff? Because we just want UNC32 GFP. Well, we can cross UNC32 GFP worms with all of this other stuff in the genotype with a wild type animal. And this would make all of this heterozygous. Then in the self-fertilizing progeny, we can select for worms which just have UNC32 GFP. And we could do this likely by looking at the fluorescence of this orange fluorescent protein, where we want the worms to be not orange. We want them to lose this cassette. And then we'd likely genotype for UNC32 GFP, ensuring that we get homozygous animals for the insert. So at the end of this, the genotype after outcrossing and homozygosing, we would get UNC32 GFP alone. And the phenotype now, because we've crossed out the mutation in UNC119, would be non-UNC. So that's what we did this semester. We designed the reagents for a CRISPR-Cas9 insertion of GFP into UNC32. We injected it into worms which expressed Cas9, and those worms were UNC because they lacked functional UNC119. In the GFP insertion, we also included a floxed copy of an UNC119 rescue within a GFP intron, which then rescued the UNC phenotype, making the proper insertion, worms with the proper insertion, non-UNC. Then we removed the UNC119 rescue by heat shocking the worms to induce expression of Cre recombinase, which acted on the floxed UNC119 rescue to remove that rescue, then leading to progeny of heat shocked worms being uncoordinated. Then we crossed out the UNC119 mutant and the accompanying transgenes to, in the end, get worms which have a GFP tag on UNC32 that are also wild type movers, non-UNC. Okay. I hope that made sense. Okay, so now I'm just going to do a quick example of how to design a CRISPR experiment using a gene that I recently tagged. So I'm just going to share my desktop. So the first thing that I would do is to go on Wormbase. This is the main page of Wormbase. And I'm going to search for the gene that I want to put an insert within. So in this case, tofu4. And this gives me some information about the gene. It's not very well characterized, so there's not a lot in the description here. I'm going to look at the gene itself within the genome. I see there's just a single isoform. And now I want to get the sequence of the gene. So I'm going to go down to sequences. I'm going to click on this view sequence button. I know one of the questions submitted was when to use the spliced versus the unspliced version. And when designing a CRISPR RNA, you would always use the unspliced version because that includes all of the sequences within the DNA. And if you use the spliced version, you might be missing something, particularly when you're designing the homology arms of the repair template. If you don't include upstream and downstream even, you might not have the full sequence of DNA required to make the proper repair template. Also, Wormbase has a very specific system of labeling their sequences when you copy them from Wormbase. So in this TAN, this is upstream sequences. In gray, this is the UTR. And then in capital is everything that's a coding sequence. So capitals are the exons and lowercase are the introns. If someone were to give you a sequence of a gene, just like this, and ask you where the start and stop codons are, the capital letters are always the coding sequences. So you know that the start codon of this gene will always be those first three nucleotides that are capitalized. So the first three nucleotides, ATG. If you didn't get the full sequence of the gene, this might not be true. So if I gave you only this portion of the gene, it wouldn't be the first three capitals because it's not the full gene. But if you were given the full gene, the first three capital nucleotides are the start codon that's used for the specific protein. And similarly, the last three capitals are the stop codon. So this specific worm-based page is telling me that this start is used in the translation of this protein, and this stop is used in the translation of this protein. Then all these gray sequences, they're the untranslated regions, meaning that they are present in the RNA, but they are not translated in the protein. So then I take this and I copy it. I've chosen the unspliced with upstream and downstream sequences. And I'm going to open it in SnapGene. It's similar to ape. And now I'm going to find where I want to insert the tag. For this specific gene, I had seen that other people had put tags on the C terminus of the protein. I wanted to insert what's called a flag tag, which works as kind of like a molecular handle. You put a recognizable epitope on your protein of interest, and then you can use antibodies which recognize that flag tag. And you can use these antibodies to either specifically purify your protein of interest, or you can do immunofluorescence, which is a way that you can see where a specific epitope tag is within fixed tissue. These are very useful for protein analysis. So I want to add a tag in the same position as other people have added other tags, because that tells me that if they made a functional fusion protein at the C terminus, that's probably the safest place for me to put my tag. So here I found the last three capitals in the file, meaning that's the stop codon. I'm going to add a feature. This is the stop. And then I'm going to find the PAM that makes a cut closest to the insert site, which the insert site I would want is directly 5' of the stop codon. So let's see. It's not looking so good, but this is a PAM, so NGG. I'm gonna label this features. Let's make this a PAM. And this would induce a cut right here. So 3 nucleotides upstream of the NGG PAM. And this is 20 base pairs away from my preferred insert site, which is not great, but I don't have so many other options. There's this CCN PAM here, and that would cut 3 nucleotides in this direction. So 3 nucleotides downstream. And that would be 21 nucleotides from my desired insert site. So I go with this as the most optimal PAM. And then when deciding the CRISPR RNA sequence, it would be the 20 nucleotides upstream of the PAM. So this is my CRISPR RNA, this is the PAM. My insert site is here. right before the stop codon. So then to design the repair template, I would first add in my flag sequence right before the stop, and then I would mutate the PAM or the CRISPR RNA, and then I would use 60 base pairs upstream and downstream of my most recent changes. So let's see what I ended up doing. This is the file for the reagents that I ordered. So I used the same PAM we recently identified, and the CRISPR RNA sequence is here. However, when I designed the repair template, I was unable to mutate the PAM without disrupting the protein sequence. So I induced some silent mutations. These are the lowercase bases here. I did 3 bases mutated, which should prevent recutting by preventing binding of the CRISPR RNA. I added in a linker sequence, which separates my tag from the protein, and it's kind of floppy. So by putting some physical separation between the tag and the protein, I am giving the best odds of having proper folding of both of those proteins without interfering with each other. So I put my flag tag and a linker right before the stop codon. And then I designed my repair template to have a certain amount of distance from each most recent change. So it'd be, in this case, I did 55 base pairs from my insert. This can be tweaked in different situations, but for this course, we'll stick to 60 base pairs on each side. Then I designed some genotyping primers. My reverse primer is this bottom strand here. So it's the reverse complement of this region, this top strand. And it is 3' of the insert site. And my forward primer is 5' of the insert site. And when using, also when I design these primers, it's important that they have similar characteristics to each other in GC content and annealing temperature, so that when I run the PCR, it works properly. If your primers are very different, it's hard to find an annealing temperature that is optimal for both of them. So the sequence that is between these primers and includes the primers is 500 base pairs. So if I have my flag tag insert, I'll see a 500 base pair band on a gel after doing genotyping PCR. And the tag and the linker is 81 base pairs. So without that insert, I'll see an 81 base pair lower band. So I did this injection and I screened a bunch of worms, and I saw some that had a heavier band. And I purified my PCR product using these 2 primers and sent it off for sequencing to make sure that the insert is correct. So this is how I generated a flag tag protein in something that I'm interested in studying its function. So I hope this was helpful, and good luck on your exam.